under aegis of star DBT status. May I now call upon Dr. Sejal Rathod, Head, Department of Microbiology and Coordinator for Biotechnology, to address the gathering.
Good evening, everyone. So yes, on behalf of KC College, Department of Biotechnology and Microbiology, I welcome our dear principal ma'am, who's always encouraging and supporting us, Dr. Nuruddin Unchwaniwala, who's the speaker for the day, heads of the departments, teachers, and all the student participants. So this webinar is second in the series of where we have called our alumni, been there and done that, where we request our alumni to share their journey that they have traveled and the destinations that they have reached currently, which will help and inspire our current students and also the students elsewhere, other places, to follow their paths, to follow the footprints that they have left. I hope this seminar or this webinar is going to be very helpful, and it's going to inspire all the participating students, and we all will be immensely benefited from it. Yeah, over to you, Rajita.
Thank you, ma'am. A leader is one who knows the way, who goes the way, and shows the way too. We at KC consider ourselves extremely fortunate to have with us one such dynamic, encouraging, and the ever supporting principal, ma'am, Dr. Hemlata K. Bagla, Head, Nuclear Radiochemistry Department, and also the Senior Dean of Science and Technology, HSNC University, Mumbai. May I now call upon Principal Ma'am for her opening address.
Thank you, Dr. Rathore, and thank you, Dr. Rajita. It's I'm delighted to be with you all always. Every evening you call me. I don't know how you are able to connect so many virologists in the last 5 months. So it's a delight to be part of this program. So we have with us in this digital room, Dr. Nuruddin Unchwaniwala. We have Dr. Sejal Rathore, who is head of the Department of Microbiology and coordinator, Department of Biotechnology. She wears 2 hats. Other teachers of the department. We also have our former heads of the department, Mrs. Prabha Padmanabha, and I'm given to understand Dr. Jaya Asrani and other heads will join very soon. Vice principals of KC College, dear students, and all dear participants. I wish you a very, very good evening. It's wonderful that you're part of this program. It shows that your commitment to be part and parcel of this program, which is organized by KC College, the international webinar on hiding in plain sight, positive RNA virus replication compartments. Friends, first of all, let me tell you all, it's always a pleasure when an institution is visited by, whether physically or digitally, by alumnus, brilliant alumni who, by their persevering acumen, are contributing to research of high caliber in the field of sciences, and all the more now in the field of virology. I'm really glad to welcome Dr. Nuruddin Unchwaniwala, an alumnus of 2006 batch. Welcome, Dr. Nuruddin. I find it very awkward to call you Dr. Nuruddin. I'll just say, because you being a student, I just feel like calling you Nuruddin, if it's fine with you. Though you are a source.
That's better. I'm not a real doctor, so just call me Nuruddin.
And dear friends, let me tell you that currently, of course, he'll be introduced in detail what I know about him, that he's currently pursuing his postdoctoral research, and he's a postdoctoral research associate at Margaret Institute of Research, University of Wisconsin and Madison, USA. So it's really, really a pleasure to have him with us. And what I know about him is, by virtue of being a virologist himself, he specializes in the field which he's going to talk about today, is about positive strand RNA viruses. And I believe his exposition to this particular field will provide us many insights about the subject, which he's going to talk about. So, and his persistence in developing his research while dealing with viruses, I felt like listening to him today. And he has worked with dengue, Zika, hepatitis C, and also works with FHV, which is my personal interest to know more about it, is flockhouse virus. I don't know how much he'll be able to touch upon, but his work is also related to a role in the formation of RNA replication complexes. So I'm sure, Nuruddin, that you'll continue to work as you're continuing to do research in this field and finding novel drug targets to restrain the spread of positive strain RNA viruses. I'm sure you'll continue to work in that area. My best wishes to you. Thank you once again for joining us today. It's a real delight to have you on, Ms. and especially when you're talking about this such a wonderful, very relevant topic today. And as I said, before we started this event, I was talking to head of the department and the teachers that we realized that last 5 months, this duration of pandemic all over the world, we have all have realized very well now how much we need virologists. So it is one of the efforts by Department of Microbiology and Biotechnology to tell our students, not only KC students, but students across India who are participating from wherever they are part and parcel of this program, that we need virologists today all the more. And we really thank Dr. Nuruddin to be being one, for being one. As a development of Covaxin is in progress, I'm sure that the likes of Dr. Nuruddin will surely prove to be the saviors of humankind. So this is the era of, I think, virologists, and they are the saviors. So it's really, really feels good that in these difficult times, a brilliant scientist are our hope, and the time has come that science was taking a backseat couple of years, and we educationists were finding it difficult to encourage students to take up the science. And they thought once upon a time science to be a student used to just, if you have higher percentages, they used to flock for the sciences. But nowadays it had last 2 decades, it was the era of commerce and the professional courses. But I think this time after this pandemic, I really hope that students pick up this subject, and especially microbiology, biotechnology, and they should realize that this is the era for them too. And they need to work very hard in this direction to reestablish our ease and pace of life. And today I'm reminded of, being a nuclear chemist, not a virologist, not a microbiologist, a biotechnologist. And it is such a fantastic thing that being from nuclear chemistry, I'm made to speak on RNAs, DNAs, genomes, and everything. But I'll quote my role model, Madame Curie, Marie Curie, who once rightly said that nothing in life is to be feared. It is only to be understood. And now is the time to understand more so that we may fear less. So indeed, it's our scientific temper that impels us to inquire and understand more and more and more. Friends, in order to understand the causes and effects of several infections, it is very, very necessary to understand how plus RNA viruses establish their replication niche. And because what I know is the peculiarity of this plus viruses, though I may be from nuclear science field, but I've been teaching over the last 30 years medical chemistry, where HIV and handling different viruses was very, very important part, or pharmacodynamic agents, chemotherapeutic agents. And we realized when talking about this, that plus viruses is their RNA viruses. The peculiarity is that they the ability to seize host intracellular membranes to generate replication compartments, or what we call RCs, which contain viral RNA and proteins, and along with several host proteins and lipids. And that create a conducive environment for RNA replication. So one of the important features, I think, of RC is that it is also forms a barrier between viral RNA replication and the cytosol, which contains innate immune sensors and RNA degradation machinery. So I think it goes without saying, therefore, that the research value of replication compartments is immense in finding myriads of drug targets. So I think this talk that we are having today with Nodoviruses is very, very important for all aspiring virologists and budding microbiologists, and even chemists, all the more for chemists too. So I'm no expert in this field, but I'm looking forward to hearing from you, Nodoviruses, because I know that is going to really interest all the microbiologists who are watching you today or listening to you today. So once again, thank you so much for giving your time and connecting to us from different parts of the world. And also, I would like to congratulate Department of Microbiology, each and every one, and Department of Biotechnology and the leading lady out there, Dr. Rathore, for really making me learn so many things during this pandemic. And you are all helping me to evolve and increase my knowledge quotient by introducing me to our alumni. It's a joy listening to our alumni and learning from students. Thank you so much. So I hope that you all have wonderful time and a lot of knowledgeable and insightful evening today. Thank you so much.
Thank you, ma'am. Thank you for this. Thank you for your kind words. And as ma'am rightly said, the importance of positive sense RNA virus, and even coronavirus falls in that one such positive sense RNA virus. Yeah, thank you, ma'am, once again. It's a pleasure for me to always introduce the host speaker for the day. And it's a prouder moment for me, especially when the speaker is your alumnus. So we have with us today Dr. Nuruddin Unchwaniwala, who's completed his PhD in cancer biology, University of Wisconsin-Madison, USA. He completed his bachelor's in microbiology from Casey College in the year 2006. Nuruddin is currently working as a postdoctoral associate at Mogridge Research Institute, Madison, under his advisor, Dr. Paul. Some of his studies include studying flockhouse virus. He has even solved a high resolution cryoelectron microscopy structure of membrane associated viral replication protein complex and even provided a new model for positive strand RNA virus genome replication. He has deciphered the conformational transition states of viral replication protein from immature to mature state during genomic RNA replication. He has designed a highly active in vitro viral replication system to study kinetics of RNA synthesis, the structure of viral replication complexes. He has engineered he has worked on engineered hepatitis B virus labeled with fluorescent proteins to track viral replication in cells using fluorescent microscopy. His work has gained novel insights into subcellular localization of hepatitis B virus core protein, which has led to new drug targets to eradicate the chronic hepatitis B virus and understand its association with liver cancer. He even established the first single RNA fish method to study hepatitis B virus. Not only that, he has worked with Novartis nucleic acid testing platform for viral nucleic acid biomarker detection, which has significantly reduced viral detection window period compared to ELISA. He has established and led the nucleic acid testing labs at two hospitals in Mumbai, India, Hinduja and KM Hospital. He has several publications to his credit in international reputed peer-reviewed journals such as Proceedings to the National Academy of Sciences and the Journal of Virology, to name a few. He is also working as a regional editor, member of Review Board for Vaccines Journal, as an independent for journals, and have peer-reviewed more than 30 articles. He's even a member and volunteer for American Society for Virology. He has won several awards and honors, as ASVL Award for American Society of Virology, the best presentation at international meeting on the molecular biology of hepatitis B virus at Oxford, UK. He even won poster presentation award at our very own Casey College at National Conference on Recent Advances in Biologic Sciences in the year 2008. He has several conference presentations, many places, just naming a few: American Society for Virology, Modrich Institute for Research. It's a pleasure for have Nuruddin with us. Thank you, Nuruddin, for taking time and being with us here. May I now welcome you to please start with your talk.
Absolutely. Wow, yeah, that was some introduction. Thank you so much. Um, let's see. Hopefully everybody can see the slides well. Okay, so as the principal ma'am just said, I'm going to talk about positive strand RNA viruses and how they form these replication compartments in cells, almost hiding in plain sight within the host cell that they've kind of invaded. And my hope is that I see there's a lot of participants today, and I'm hoping I can explain things well enough that people who are not in microbiology or chemistry background can still hopefully get something to take away from this. So I apologize ahead of time if I'm too complex at times and I'm not breaking down complex ideas well enough for you to understand. Just beginning, how we think about storage of genetic information is the central dogma, where you have genes that are encoded within double-stranded DNA, and that becomes RNA and then proteins as the flow of information goes in our bodies and in other eukaryotic cells. But viruses have devised ways to use all kinds of nucleic acid intermediates to store genetic information. And hence they're divided into 7 large classes based on what kind of genetic information they store. So class 1, for example, would be double-stranded DNA virus. But there are other viruses that store information in either single-stranded DNA, RNA, and all kinds of forms. And the class that I personally focus on in my work is positive strand RNA viruses, which is basically this class number 4, which stores the genetic information as single-stranded RNA, converts that to minus-strand RNA, and then back to mRNA, and then that makes all the proteins. So if you'll appreciate that these kind of viruses never use DNA in their life cycle. All the genetic information is all stored in a molecule called RNA. And this particular family includes major human pathogens. Some examples are Hep C, dengue, Zika, and SARS and MERS coronaviruses, including the current pandemic SARS coronavirus 2 that we are all living through. And controlling these viruses requires understanding how they spread and replicate. And that is basically the goal of my studies. So what is a general replication strategy of positive strand RNA viruses? So you can imagine a virus infecting a host cell. And remember this, since the virus never uses DNA, it never explores the nucleus of a host cell because it doesn't have a need to use DNA or go into the host nucleus. So there's only a single RNA molecule that goes into the cytoplasm. And since it is plus strand, what it means that it is a messenger sense RNA, it can directly translate proteins. And that is exactly what happens next. The RNA gets translated to viral proteins, and both the viral RNA and the proteins are then trafficked to a central host membrane to modify the host membranes to form what is called as RNA replication compartments or replication complexes. I use that interchangeably and they mean one and the same thing. They are these modified host membrane structures that house both the viral RNA and the proteins. The kind of host membrane these viruses use depend on which virus you're looking at. They could be either ER membranes, Golgi, mitochondria, peroxisomes, or plasma membrane. So anything in the cell that is made out of lipid membranes is potentially explored by different viruses to form these compartments. And there's only one major role of these compartments is to replicate the genomes. So the viral polymerase, which is the enzyme that converts the single-stranded RNA to first double-stranded RNA and then back to a lot of plus-strand RNA copies, is housed within these replication compartments. And as you imagine would happen here, the outcome of such a replication compartment is a large number of copies of genomes, which you see here. And these genomes are then packaged into new virus particles and they're released from the cell. So that kind of completes the life cycle. And then the viruses move on and infect a new cell. And it is these replication compartments that is really the focus of my study. We as a group wanted to know how these structures are formed and how they function so that we can better understand how to stop these replication compartments from forming and functioning. So one obvious question is, why would the virus spend all this energy to form replication compartments? There are various ideas in the field and why viruses have evolved, positive strand RNA viruses have evolved to do this. One is efficiency. It allows the viruses to concentrate all the important viral factors on a host membrane. So they're not diffused in the cytoplasm everywhere. They are aggregated together on a membrane. So it kind of enhances the functionality of these viral proteins and increases the amount of viral RNAs that can be synthesized. The other goal of this is to segregate the plus strand RNA from both translation and replication. So because the viral RNA is also messenger sense, if it's just freely floating in the cytoplasm, how will the virus be able to distinguish what is a translational template versus what is a replication template? So if these RNAs are housed, protected inside a membrane, their sole function then becomes to replicate the genomes, and the ribosomes can never access these RNA molecules. The last one that actually in the introduction itself, the principal ma'am referred to, is protection from host defenses. Uh, if the viral RNAs, especially double-stranded RNA, is a strong activator of innate immune responses. So if it's freely floating in the cytoplasm, the host cell can sense it and induce a strong antiviral response. So to protect so the virus tries to protect that from happening, and hence it tries to kind of hide behind these replication compartments so that it can efficiently replicate the genomes and at the same time not be detected by the host innate immune responses. And it turns out that this formation of replication compartments is a conserved strategy across all positive strand RNA viruses. And this includes a large number of viruses. And hence it becomes a critical thing to understand in terms of structure and function so that we can better target this with like new drugs. So what have people done about this? So these structures are generally really small. And what do I mean by that? So the diameter of a typical replication compartment is only 50 to 100 nanometers. That's too small to be seen by light microscopy. And hence there's a specialized technique called electron microscopy that is used to see these structures, because using electrons as an optical source will allow you to resolve structures that are much smaller than what light microscopy allows you to do. And here are some examples of 3D reconstructions of replication compartments from many different viruses, which form these vesicular structures, spherical structures inside cells using a variety of different membranes. So some examples I'm showing you here are hepatitis C, and you can see these golden spherical structures. These are completely reorganized endoplasmic reticulum membranes inside infected cells. Similarly, in SARS coronaviruses, you can see these giant double membrane vesicular structures. And of course, these are all false colored to kind of highlight the nature of these. In electron microscopy, you generally see just a grayscale image. And these are then 3D segmented to kind of show or highlight the different features. In dengue, the same thing. Flockhouse virus is something our group has studied for many years now, and it forms these similar vesicular structures on mitochondria. And also for West Nile viruses, it uses ER membranes to rearrange and then form these structures. So we can immediately see how dramatic these rearrangements are. ER membranes and mitochondria don't look like this. They're modified extensively by these viruses for a sole purpose of hiding the genomes and replicating the genomes. And hence, understanding how these look and function is kind of very critical to understanding how the viruses kind of hijack cellular machineries. Something that is done in the classical EM work or tomography work like this is there's a lot of sample processing involved, and there's a lot of chemical fixation involved. So even though you see these membranes really well, you cannot really see viral proteins or viral RNA. So you can notice that these structures are kind of empty, and that's just due to processing. So you kind of lose information on the associated proteins and viral RNA. In contrast, there are a lot of groups that do fantastic work with purified proteins. And what I'm showing you here is crystal structures that have been solved for proteins that are associated with genome replication in positive strand RNA viruses. So they basically have purified the proteins involved and solved the structures. And these are extremely informative in terms of understanding how genome replication is working at a molecular level. But with purified proteins, you kind of completely miss the information about the host membranes that you saw earlier. So we as a group wanted to kind of bridge this gap and provide not only information about how membranes are modified, but at the same time also show how the proteins that are involved in genome replication are housed or are present at these replication compartments. So kind of put both together in some way. And the way we do this is by using a technique that will help us provide kind of native structural views of each component of a replication complex, or proteins, membranes, and RNAs. And we use Nodoviruses as a model system, and specifically the virus in this family that we use is called Flockhouse virus. So just tell you a little bit about nodoviruses. And why we use them as a model system is they're really small genome viruses. They encode for only 3 RNAs. The total genome size is only 4.5 kilobases. So it's really, really small, even in terms of a virus. And it only encodes for 4 proteins. So it makes our life a lot easier studying these viruses because we don't have a lot of viral proteins to target. It's an extremely low risk pathogen. It is not infectious to mammals. Nodoviruses infect fish and insects. So we can work with extremely infectious wild type viruses in a lab setting without having to worry about getting infected ourselves. The RNA replication of these viruses is functional across many different cell types. So it helps us use a variety of different cell types like plant cells, mammalian cells, yeast, C. elegans, and others to kind of dissect the molecular functioning of different viral proteins and structures. And the replication compartments of these particular nodoviruses form at the mitochondria. And you'll know in a little bit why mitochondria is very useful. As an organelle inside a cell, mitochondria is extremely easy to isolate from cells. And hence we can lyse a cell, get all the mitochondria, and image the mitochondria in isolation. And that has actually led to a breakthrough in terms of understanding these structures a lot more than anything else has. So I'll show you in a moment what these isolated mitochondria look like. So the technique we use is called cryoelectron microscopy. At its very core, it's a very, very simple technique in terms of sample preparation. The cryo comes from the fact that we prepare our samples and image under cryogenic conditions. And what I mean by cryogenic is liquid nitrogen temperatures. So we are talking about minus 80 to minus 190 degrees Celsius. And the idea behind such a technique is that if you put your sample on a mesh, like things shown here, and if you very quickly freeze it under liquid nitrogen conditions, which is called plunge freezing, you form a very thin layer of amorphous ice along with your sample. And this really quick freezing, which forms amorphous ice, is an extremely good substrate for doing electron microscopy imaging, where you don't have to use any kind of fixative or any kind of other chemicals to fix your sample. And hence, you can retain all the native information of the sample intact while imaging. And hence, you can actually see all the detailed molecular structures, which would otherwise be disturbed if you had used any chemicals or any other fixative conditions. So you basically have a grid like this, and the grid has holes. And what you see here as tiny gray squares is basically your sample that is suspended in a thin layer of amorphous ice. And then you take the sample over to a cryoelectron microscope, which uses electron beams as an optical source, which is then you basically shine that on your sample. And through transmission electron microscopy, you get a diffraction, you get a transmission pattern on a camera, which you then record, and you can get very high resolution information about different structures in your sample. An additional thing we do on top of this is called cryotomography, where you basically take your sample. So if you imagine this blue protein complex here as your sample, you tilt the stage on which your sample is sitting, through which the electron beam is going perpendicular to, and you can imagine your sample stage tilting back and forth, and that allows you to get 3-dimensional information on your sample. And what you can do is apply a technique called back projection for these tilted image series to then convert them to a serial stack. So imagine a movie of serial images running as a Z stack going through your sample. And then you can basically that allows you to get 3-dimensional information on your sample, which you otherwise will not get if you just took a single image. A single image is just a 2-dimensional image, but a 3D image is very important to get the whole 3-dimensional information of these samples. So this is the core technique, cryotomography, that we use that has led to all these breakthroughs in terms of identifying these structures. So what I'll show you now is mitochondria from a nodoviral infected insect cell that we isolated and plunge froze and did cryotomography on. So remember, again, what I'll show you now is a movie that will run through the different Z series of the sample and show you the 3-dimensional view of the whole thing. So what you see now is a single mitochondria, and you will also be able to see the replication compartments I was talking about. So as this movie is running through, you will see a lot of these circular structures at the outer membrane of the mitochondria. And these are only seen in infected cells that are housing these things called replication compartments. And if you look closely at a single section of such a movie in kind of a more zoomed in view, I'll be able to highlight some of the different features of what the replication compartments look like. So the red arrows are highlighting the outer membrane, which is running horizontal to the slide right now. That is the outer membrane of the mitochondria. The blue arrows are showing you the balloon-like structures or vesicles or replication compartments that the virus is forming using the outer mitochondrial membrane. What you're seeing in black arrowheads is fibrils leaving a single replication compartment. So what you're basically seeing is most likely a single molecule of RNA emerging from replication compartments going to the cytosol. Kind of highlighting how replication works at these replication compartments. And what I'm showing you in white arrowheads is an extra bits of density, what we call, at the neck of where the replication compartment meets the outer mitochondrial membrane. And I'll come back to this a little later. So keep this in mind that there is some extra bits of densities right at the neck. So the other thing that some of the previous people in my group did was they wanted to understand if some viral proteins are present at these replication factories, specifically the viral enzyme that is needed to replicate RNA. So, and that particular single viral protein is called protein A for nodoviruses. And this technique that I'm showing you here basically labels proteins with an antibody which has a small gold molecule on it. So gold particles are extremely electron dense. And because of their high electron density, they diffract a lot of electrons in electron microscopy, and they're very easy to identify. So hence, if there's something that is very electron dense, it will show up as a very dark black spot. So what you're seeing here as these dark black spots are gold nanoparticles. And the gold is bound to an antibody that recognizes a viral protein. And hence, you can clearly see here that on a mitochondria that has these replication compartments, you can densely label them with antibodies that are specific for this viral protein called protein A that is found here. So right away, we knew that these replication compartments are not only viral, but they have a very specific viral protein called protein A because of the specific labeling. So what exactly is protein A? The nodoviral protein A is a very large single non-structural protein. It is about 1,000 amino acids long and has various functional domains going from the N-terminal end to the C-terminal end. And more than 10 years of work in our group has established a lot of different features about this protein. It is a protein that self-interacts, that is, it multimerizes into a larger complex. It is known to be membrane bound, which makes a lot of sense because it has to, the virus is functioning at a membrane by modifying membranes. And the two main membrane binding regions are the transmembrane domain at the N-terminal end, and the other domain called iceberg that is in the middle of the protein, shown in green here. And it also has two separate enzymatic domains. One of them is the polymerase, or the RNA-dependent RNA polymerase, that will replicate the viral RNAs to then make more viral RNA copies. And the other is capping, which is shown here in pink. Capping is a process that happens on plus-strand RNAs at the 5' end. And the capping allows the viral RNA to be more stable and then become competent for translation. So coming back to this image that I showed you earlier, and remember how I told you to kind of keep this in mind, the extra bits of densities shown in white arrowheads. And this is what I'll get back to now. This is kind of the core part of the study that we are doing. We saw these additional densities, and in cryotomography, there is a technique that you can use to further enhance what you're seeing by averaging multiple structures. So even though what you're seeing here looks very fuzzy and not very clear, but if you were to take boxes of chunks out in 3 dimensions and take individual structures like these out and average them together by a technique called subtomogram averaging, you can actually enhance the structural views of these particular densities that you're seeing if in case they are homogeneous, if they all look very similar, then you can average and get a much higher enhanced structural view by subtomogram averaging. And that is exactly what we did. We took thousands of these different structures individually and put them together by subtomogram averaging and averaged. And here's what we saw. And it was kind of really striking what we saw, because it was a highly defined symmetric structure that sits at the neck of these replication compartments. So now, as I play this movie, I'll kind of show you in 3 dimensions what the structure looks like. It has 12-fold symmetry. So as I rotate the structure around and top, you can see how it has 12-fold symmetry across the central axis. This is made up of largely just the single viral protein A. It is a gatekeeping protein complex that kind of guards the entry and exit port of a replication compartment. And basically, it controls viral RNA replication because protein A has the enzymes required to do so. If we want to look more closely into the structure, I want to just kind of get across this idea about thresholds. It turns out that when we look at these structures in the native state, we can play with the electron density of these structures and deduce what is protein and what is membrane. So if you remember what I said earlier, in all the other studies that were done before, you could either see just the membrane or you could see just the proteins. But we were trying to see all of the things together. And that's the nice thing about such kind of a study. If you look at things in high threshold or low threshold, you can actually define what is protein and what is membrane. So on the left, here is a high threshold structure, and you're largely only seeing protein. When you play with the electron density and lower the thresholds, you can see protein plus the surrounding membrane. So to try and show all of these things together, what I've done here is I've taken the low threshold structure and made it a transparent white. And I'm now going to put this high threshold structure inside, and I'll do a cross-sectional view so that you can appreciate how the membrane wraps around a structure like this. So now what will happen is I'll take off sections from the front and the back. And what you're seeing now is in blue, the protein replication complex crown structure surrounded by the lipid membrane. And what is really striking here is that even though the replication compartments, the vesicles underneath here, are of many different sizes, they go from 30 nanometers big to 100 nanometers big. Even though that's the case, the lipid membrane that surrounds the crown in the vicinity of this protein structure is highly constrained. And because we are seeing things at such a high resolution, you can actually even see individual lipid bilayer leaflets. So if you're familiar with what a lipid membrane looks like, it has 2 layers called the lipid bilayers with polar head groups and the fatty acyl chains in between. Because the polar head groups are higher electron density, you can see a single lipid membrane as 2 bilayers running parallel to each other. And I've kind of shown that with a dotted line here to kind of guide you how this membrane is curving. So what you can appreciate here is how strong the forces are generated by a single crown complex in blue here to significantly bend the lipid membrane to induce curvature that is required to form a balloon-like structure or a vesicle that sits underneath it. And this is where the viral RNA is hiding beneath and replicating. So it's a beautiful structure with very high complexity and very high, it's a very intimate structure that is formed along with the membranes in cells. So even though we knew there's protein A in the structure, we did not know how protein A is organized. So we can see a high resolution structure, but we don't know where the N-terminal end and the C-terminal end of the protein is. So we further mapped how protein A is organized in the structure. So to map this, we used a technique called his tagging and goal labeling. So again, I'm showing you a linear map of protein A, which is about 1,000 amino acids. And since we wanted to basically know where the polymerase is, we tagged the C-terminal end of this protein with a his tag. So his tag is nothing but 6 residues of histidine amino acid. And the nice thing about a his tag is that it coordinates a nickel ion by chelating it. And you can use that chemical property of his tags to specifically label a certain section of a protein with extremely electron dense gold particles. So in this case, we have gold particles that are bound to nickel, and nickel will be coordinated by a his tag. So when we take our samples and label them with gold, we'll be able to see where the gold is by electron microscopy and hence know where the C-terminal end of protein A is in our structures. So that's exactly what we did. We made genetically modified viruses in lab, infected cells, and then isolated mitochondria. And then these mitochondria were treated with 5 nanometer gold particles. And then we plunge froze and did cryo-ET imaging. So on the left is what a wild type virus sample looks like. So there's not much gold here on the surface of mitochondria. But in contrast, when you use a modified virus with a His tagged protein A, you start seeing a lot of gold clusters showing up right where the replication compartments are. And if you were to zoom in into a few of these, you can actually individually see gold particles on top of these replication compartments. So we were able to specifically label protein A at the replication compartments. So we went back to our gold standard technique of subtomogram averaging, where we extracted a lot of these different crowns from these images and did averaging. So if you average just a small number of crowns with no gold, that's the structure in blue. And if you were to mix in these blue crowns with gold label crowns, you can see there's a large kind of fat kind of density that shows up at the top. And if I were to overlay these two structures together, you can see there's a lot of extra density that is showing up right at the top of the crown complex or at the apex. So since we know we were labeling the C-terminal end, that kind of already told us that, okay, so the polymerase, if the gold is showing up at the apex, then the subdomain here at the top, which we call the apical lobe, is possibly the polymerase domain. So we use some kind of a correlative deduction after this to figure out whether that's the case. So since the polymerase structure is not known through any kind of crystal structures, we used software algorithms to figure out the predicted crystal structure of a polymerase domain. Since a lot of RNA viruses make polymerases, the structures of polymerases are heavily conserved. So we were able to deduce what the nodovirus polymerase would look like. And that's the predicted structure I'm showing you right now. And we asked ourselves a question: can we take this predicted structure and try to fit that into the top subdomain of the crown? And does that fit? That's the basic question. It's a very simple question. When you put that structure in, does that seem likely to be a good fit? And that turned out to be exactly the case. The apical lobe here at the top of the crown complex fits very nicely with the predicted structure of a polymerase domain. So that tells us that our His tag labeling and the structure fitting, which we call rigid docking methods, is showing us that the polymerase of the virus sits right at the apex of the crown, and hence the remaining N-terminal portion of the protein A is all down here, forming a single copy. So the current model of what we think a crown complex looks like is something like this, where I've colored these alternate white and blue so that you can appreciate the 12-fold symmetry and how there are alternating units of similar protein coming together. So there are these 12 parts that come together to form a crown complex, or 12 copies of protein A, and they're arranged end to C basal to apical. What it means is that the membrane is down here, and the apex of the crown complex is right there. And in yellow is the identified polymerase domain, and the N-terminal half, which makes very strong membrane connections with transmembrane domain and iceberg, are down here in 2 separate connections that you see per molecule. And hence there are 12 such molecules surrounding a crown complex, which significantly modify the membrane underneath. So here's the model of how we think everything is working in the big picture of RNA replication. So you initially have a crown complex sitting on a mitochondrial membrane, or in other viruses on any other membrane like ER, Golgi, or peroxisomes. And the viral RNA template comes in, and that comes in contact with the viral polymerase, which immediately starts synthesizing a double-stranded RNA. And remember, right now, there is no membrane modification happening. There is no vesicle underneath. The vesicle only forms once the RNA is replicated to form a double-stranded RNA molecule. And you can imagine this like blowing up a balloon, where this newly synthesized double-stranded RNA is kind of feeding into a balloon that is kind of blowing up. And that is what you see as a vesicle underneath. once the double-strand RNA is fully synthesized. So that is only one half of replication though. First half is making double-strand RNA. The second half is called transcription, where you need to make now new plus-strand RNAs. So that is where things get a little complicated, because the polymerase we have identified is way at the top here. So how does this polymerase replicate this double-strand RNA that is really far away in terms of distance? Even though the distance is not too large in real life, but in molecular life, this distance is really long. It's about 9 nanometers. So we suspect there could be another copy of protein A inside the vesicle, or there could be a conformational change that brings this polymerase down into the hole and contacts with the double-stranded RNA to then synthesize the new plus strands. And you can imagine the plus strands then leave the replication compartments, and you can see these as like fibrils coming out, which I showed you earlier in some of my cryo-EM images. You could see these strings of fibrils leaving replication compartments. So that's the current model of how we think everything is working in terms of the identification of this crown complex and everything else. So lastly, you might be thinking, is this similar for coronaviruses? Do coronaviruses have a similar structure like this? So I'll show you just now some false colored models. So this is what we discovered as a group in Nodoviruses, where you have a replication vesicle in white, the RNA inside in red, and the crown complex I just showed you in blue on the top. It turns out just like 2 weeks ago before this talk, a new paper is coming out. It's not published yet, but you can still find the preprint online. And this paper was about coronaviruses, also specifically SARS coronavirus 2, that is an ongoing pandemic virus. And a group through collaboration between Germany, US, and Netherlands, we're trying to figure out if such a structure does exist also in coronaviruses, and indeed it does. So in coronavirus replication complexes, you see a red crown-like complex on a membrane vesicle. You see the RNA in green inside this vesicle, and in yellow is the ER membrane. So like nodoviruses use mitochondria, coronaviruses use ER, modified ER membranes to form this. But what I'm hoping you will get from this is how similar these structures look in terms of how they function and how they have a crown-like complex that kind of gatekeeps the whole structure and controls or protects it from innate immune responses. So this is why studying basic models like nodovirus is extremely useful, because you can apply the information you get from it to other viruses, including extremely pathogenic pandemic viruses like coronaviruses. With that, I would like to acknowledge everybody in my group who has been extremely supportive and great, and I'm really grateful to this to a lot of people for all of this work in my lab specifically. And my co-author on a lot of the work I do is Hong Zhan. He does a lot of cryo-EM imaging and data computation work, and kind of we work in tandem together to generate the new ideas and data sets about viruses like these. And of course, there's a lot of other members in my group who have different expertise. So I just want to point out that all of this work I'm presenting here is not just mine. It actually belongs to our whole group who contributed in many different levels. We do our imaging at 2 core facilities in the US, at the Pacific Northwest Cryo-EM Center and the Janelia Farms Cryo-EM Center. These are extremely expensive microscopes that we use to do cryo-EM imaging. So I'm really grateful to these organizations to provide us support for this. And we collect data sets in terabytes for every experiment. So we generally have to use extremely high computational power to average structures that runs for days at times before we see anything. So we do invest a lot of money in computation to do all the high resolution imaging. And of course, there are a lot of different agencies that fund this work. University of Wisconsin, Morgridge Institute for Research, and Howard Hughes Medical Institute. So all the money that comes in to do this work, we are extremely grateful for all of that. And with this, I would be really happy to take any questions or comments. I'm really looking forward to that. Thank you so much.
Niruddhan, there's a question posted just now.
Yeah, let me.
In the chat box.
Yeah, I hope I can open up a chat.
Okay, I'll read it out for you. Are there any antivirals that prevent the formation of these vesicles or the protein A?
No, not right now. So the only that's a great question. The real focus for antivirals for positive strand RNA viruses is to block the polymerase from functioning. There has been no antivirals that stop replication compartments from forming. So we are hoping that these kind of studies from our group and the other groups who are studying these will lead to molecular insights that will hopefully help us design new antivirals that actually block these membrane associated viral proteins from forming these modified membranes in the first place. So one of the good examples of a very successful drug that blocks the polymerase from functioning, which is the only class that is available right now, is for hepatitis C. There's a nucleoside analog that can cure hepatitis C within like 2 months of treatment. And that just functions by blocking the polymerase. But the problem with polymerase is that in RNA viruses, they mutate very quickly. So any drug that you have that functions right now to block a polymerase can very quickly become stale and not functional anymore. Whereas these core membrane associated compartment functions are more conserved. So anything that is very well conserved has a very high chance of being successful long term because the viruses cannot deviate too much from the core structures like that. So the hope is that these studies will hopefully bring out some molecular insights that can be used for making new drugs. I see one more question from Grace. It's about, does one vesicle contain just one RNA molecule or could it contain multiple molecules? That is an amazing question. What we think right now for at least nodoviruses is that every replication compartment has only one template. Because what we found was that the size of the replication compartment was dependent on the size of the template. And if there's a dependence on the template size to replication compartment size, we think it is most likely that every compartment has only one copy. But that being said, for coronaviruses, it turns out to be that I think there is more than one copy because coronavirus replication compartments are massive compared to nodoviruses. So nodovirus replication compartment is about 100 nanometers big, the largest. The coronavirus largest replication compartment is about 500 nanometers. So we think for coronaviruses, there is more than one template per vesicle, unlike nodoviruses. The next question from Lena is, do we know what factors determine that a particular RNA virus will use ER or mitochondria to make replication complex? There are signature sequences within viruses in their proteins that traffic them to the right membrane. So nodoviral protein A, when I showed the transmembrane domain at the N-terminal end, that is actually a mitochondrial targeting signal. So if you delete that, the protein does not go to the mitochondria anymore. Likewise, in coronaviruses and dengue and Zika, they all have proteins with signature peptide sequences that target them to a specific membrane. So maybe the obvious next question is, why do viruses use different membranes? We don't know. We don't know why coronaviruses use ER, for example. We don't know why nodoviruses evolved to use mitochondria. The only thing we know is that they all go to membranes. We don't know yet why they go to different membranes. There's a question by Vijay. I want to ask whether any controls having normal mitochondria were set up to confirm the absence of, oh yeah. That is absolutely the first thing we do is we look at just normal mitochondria. And a normal mitochondria would look like any textbook image of a mitochondria that you would see, where you see the outer membrane as a continuous membrane and the inner membrane as cristae that is very tightly organized in the mitochondria. So obviously, yeah, we do all these different kinds of controls, and that's a good question. I mean, it's always a good thing to use controls in experiments when you're trying to compare what's happening. So we obviously do that. Does the RNA virus lack proofreading activity from Adarsh? Yes, so most of the positive strand RNA viruses lack proofreading, except coronaviruses. Coronaviruses have a specific enzyme, RNA exonuclease, that is dedicated to proofread. And hence, coronaviruses, unlike other positive strand RNA viruses, does not mutate as quickly, which is good news for us because we are trying to make vaccines right now for coronaviruses. And if the viral proteins were to mutate very quickly, we would have a really hard time keeping it under control. One example I would give in that regard is influenza viruses. Influenza is not a positive strand RNA virus, it's a negative strand RNA virus, but influenza undergoes genetic shifts and drifts very quickly, and hence we have to make vaccines every year for influenza. So, but coronaviruses present their own challenges. That it more likely the challenge with coronaviruses has to do with our own immunity and how weak our immune responses are and how short-lived they are with coronaviruses. So hopefully we can come up with something as a vaccine that allows us to generate a long-term response to something that is very conserved in the virus. So next question from Suvarna is, so how viruses undergoing mutations change the organelles it infects? Great question. So for nodoviruses, the mitochondrial structures look pretty much the same, even though the viruses mutate a lot. So what that tells you is any virus that mutates to a point where it cannot effectively do the conserved function of replication compartment formation, they basically die out. So you can imagine viruses as a big gene pool or quasi-species where there's all kinds of mutants in the population. And only the mutants that can do the conserved core function survive and everything else dies. So that's how the virus the landscape changes over time. The organelles itself that they infect remain the same, and the organelles it's also look almost the same in a lot of different mutant viruses. From Ronit, I have, can the formation of replication complexes be hampered if you were to somehow target protein A? Yep, that's exactly what we're trying to work on right now. We're trying to understand how protein A is organized and how it multimerizes and try some small molecules to see what we can do in terms of not forming replication complexes. We are as a group set up to do a lot of these high resolution imaging studies. So we think we're in a good spot to try and do some mutational analysis and small molecule analysis like this to figure out how we can hamper replication complex formation. From Anaka, I have, there is no host defense to the translation of viral RNA in the host cell. It is not. And the reason for that is the viral RNA that is capped at the 5' end looks very similar to any other mRNA that your cell is making. So the host defenses are not really targeting or even sensing the viral plus strand RNA in the cytoplasm. The only thing that the cells don't like seeing is the double-stranded RNA. Because if you imagine in your normal cells in your body, there is no pathway for double-stranded RNAs to be hanging around in the cytoplasm. So anytime the host cell sees a double-stranded RNA in the cytoplasm, it gets alerted that there's something wrong. So the double-stranded RNA template is what the viruses have to protect, not the plus-strand RNA, mRNA-like looking molecule that leaves and translates to make proteins. From Nilantana, I have, what will happen if trafficking of the fibers are blocked by drug? Then what will happen within RCs with excess concentration of plus-strand RNAs? We don't know. That's a very nice curiosity question, kind of almost like the kind of work we like to do in terms of curiosity-based science. If the fibers are blocked, that could very well be studied by making very subtle mutations in protein A. And that is some of the things we're actually doing right now. We do not have a drug yet that we can successfully use to like just block the fibers from coming out. It's almost thinking about what if you took the gatekeeping molecule and blocked the hole by putting something in it. It would be really nice to have something like that. We don't right now, but that's a very nice kind of a thought experiment to think about what will happen. And my honest answer is I don't know what will happen. From Rekha, are viruses resistant to antivirals inside the replication complex? I don't know. So the pore, so if you can see the screen still, this pore, the central pore, is about 19 nanometers wide. That's pretty big for small molecules and drugs to go in. So my guess would be that if drugs were to passively diffuse through the pore like that, they would not be protected. We could still get the drug inside. Now, if there is an active process through which things are taken in and out of the pore, then the antivirals could be blocked. But we don't know exactly how the pore works yet. Like, how long is it open? When does it open? Does it only open when RNAs are coming out? So it could be a very tightly guarded uh, uh, junction. And if the junction is working like as a very tight junction, which is controlled heavily, then antivirals might have a hard time going in. What's next? From Shabir, I have, so what do you think is coronavirus able to survive in every region of the world? Is this mutation capability or protein structures or anything else? I think it's probably not mutation because coronaviruses don't mutate that heavily. I think it is the fact that the virus originally emerged in animal hosts. So it's a zoonotic virus, largely coming from bats. We don't know what the secondary animal was. There has been some ideas about pangolins being the secondary host, but that is not confirmed yet. 90% of the sequence of the SARS coronavirus 2 right now matches a bat coronavirus. And bats have been known to host a lot of highly pandemic pathogenic viruses just because how their immune systems work. And bats are mammals who fly. And if you have other mammals who can fly and cover a large region, they're able to also host viruses that can evolve significantly in a mammalian host and then pass it along to us. Hence, hanging around bats is generally a bad idea. But I think it is the fact that these bat coronaviruses have been able to jump, and it is just it's just a matter of chance that one coronavirus emerged that was capable of infecting humans and spreading extremely quickly because we have not seen anything like that ever before. So our immune systems have not seen anything like this, and the virus was able to evolve long-term in bats and jump to humans, and it is just it's a matter of chance. And I think it's not so much about mutation itself, but it's just the right combination of spike and other combination of proteins that makes it extremely contagious. From Chirag, I have nodovirus is basically a mitochondrial membrane for formation of RCs. Is there any link between assimilation of energy? Oh yeah, so I get that asked all the time, Chirag, and that's a great question. We have found that there is no real need for mitochondria itself in terms of energy. And the reason I can say that is that transmembrane domain on protein A that I showed earlier, we actually changed that to an ER signal. So we actually made the replication complexes form artificially at the ER membrane, and it worked just fine. So what that tells you is that it doesn't really need the mitochondria for energy. It only needs the mitochondria for membranes. So it turns out that the membranes are required, but not the real function of the organelle. So it doesn't care if it's at ER or mitochondria. It is just something that viruses have picked up evolutionarily as a signal, and they've found that something works, and they've just kept it. So there's no real particular need in terms of energy from the mitochondria itself. Why are there no vaccines from Karun sir? I have why are there no vaccines for dengue even after so many years? It is just that some viruses are extremely challenging, and especially with dengue, there have been efforts to make vaccines, but some of the recent efforts have actually turned unfavorably for us. Where if you give somebody a vaccine for dengue or a promising vaccine for dengue, it makes them even more susceptible to get dengue later or a different strain of dengue because of a process called antibody dependent enhancement, or ADE. That's very well studied now in the field of dengue and chikungunya and Zika, where a previous exposure to a different dengue strain makes you even more worse for the next dengue infection. So it tells you that there's something we still don't understand about a lot of these viruses, and that is why doing basic molecular biology or virology is so important to understand deeply how these viruses work. Because if you go in with half information, sometimes you learn something that you did not expect to learn, and it kind of puts you back to square one, where you have to go back to the drawing board and try to understand how these viruses work. So there have been efforts with dengue, Zika, and chikungunya to try and make vaccines. But recently, a lot of hurdles with ADE have kind of got scientists back to the drawing board and trying to understand how to target this. From Desiree, I have, do these membranes provide a favorable condition or nutrition for the viruses to grow? I don't know more about like the nutritional aspect, but these viruses need membranes and hence they need lipids. So we do know for a fact that the viruses need a lot of lipids to synthesize a lot of these viral structures. So you can maybe call that nutrition for the virus, because if the virus needs something to grow, you could think of that as nutrition as well. But so yeah, definitely they need a lot of different lipid components like PCs, phosphatidylcholine, cardiolipin, and others to kind of grow these replication compartments. From Madhvi, I have, oh, the list of questions is long. I'll try my best to go through these.
Nuruddin, you could just pick up maybe 2 or 3. And
okay,
then
let's see. Yeah, I would love to answer all of these. If you still have questions, I'll say this right away. Please feel free to email me. I'll be happy to have a discussion. It already looks like you guys are asking some fantastic questions that I really, honestly, I wasn't expecting this deep sort of insights from everybody. I'm kind of struck completely by this. Let's see. I'm trying to see what are some major questions I can answer right now. There's a very interesting question from Dhanashri. Membrane formation processes can be targeted by autophagy process. So I think the question might be about how you can redirect autophagy to kind of take over these replication compartments and shut them down. There has been some interest in doing so, but because autophagy is a completely distinct pathway, it has been really complicated to figure out how to merge these pathways together. Or are there kind of antivirals you can give somebody that will make the cells take over these replication compartments and soak them up and not allow the viruses to work? So yeah, that is something that has been looked into, but I don't think there's been much success. From Harsh, I have, do viruses identify receptors present on the membranes and select the membrane for replication? In terms of intracellular replication, in terms of replication compartments, I don't think so. It is practically a matter of just the viral proteins going to a membrane and just binding. They might be using host proteins for a function, but I don't think they use them as receptors to initially bind. I think the initial receptor binding is only happening when the virus enters the cell. So that kind of handshake mechanism that you can imagine, when the viruses are coming in and binding a host cell, and the right kind of handshake has to happen for the viruses to get in. So that is happening at the receptor level on the cell surface, but I don't think so much internally on replication compartment formation. Let's see. I don't know the name of this person, but the question is how coronaviruses evade host cell defense. Decoded, can nodovirus protein structure decode, mutate? I don't think I understand this question really well, but I think it might be about how coronaviruses are able to evade host cell defenses. At least at the replication level, it is definitely the replication compartments, because they're kind of hiding in plain sight in terms of how they replicate their genomes. But there are a lot of other viral proteins that are made, and there could be a lot of other mechanisms as how it evades host immune systems. And the thing to note here is that the kind of presentations I did here and others do, they're only answering a snippet, a really small area of viral research. And you can imagine a lot more work is required by a lot more virologists around the world to deeply understand how these viruses work. And there could be other host cell defense evasions that the virus is doing that we don't really understand yet. Let's see. From
last question, yeah.
Yeah, let's see. I think some of the others I've already answered indirectly. So the last one from Shruti I have is, what makes some individuals remain asymptomatic in spite of the virus being with the individual? That's an amazing question and extremely relevant to coronaviruses right now, where about 30 to 35% of people who get coronaviruses are asymptomatic. They're actively shedding the virus, make no mistake, but they're asymptomatic. The symptoms you get after viral infection are not so much the virus, but your immune response to the virus. So some people's immune systems are driven to make a really, really strong response to an infection in terms of cytokine responses and innate immune responses and adaptive responses. Whereas in some other individuals, the host response or your body's response to the virus is not that dramatic. So even though you are sick inside and you're making viruses, the large amount of symptoms like feeling the tiredness or lethargy and having a runny nose and sneezing and having fever are all associated with your body thinking that there's something wrong. So for some people, the immune systems are not driven or designed in a way that they should react extremely strongly to the virus. And hence they can stay asymptomatic, but in terms of viral replication and everything else, nothing is different. They have cells in the airway that are infected. They have cells that have large number of replication compartments, and they're making a lot of viruses. It's just that how their body responds to the virus is different, which makes them asymptomatic. I think that was one of the last one. I don't see more questions. But yeah,
ma'am, your thoughts, Bagla ma'am, your thoughts on it?
Hello.
Yes, ma'am.
So Dr. Nuruddin, thank you so much. Very, very informative session, and a lot many things I realized we haven't read and we don't think that is important. But today I realized, can we introduce your topic can be part of our syllabi. So, and you'll be very happy to know, that your college, KC College, is now part of the new university, which is HSMC University. Yes. So all the more, I would like Dr. Rathod to pass on the syllabi to you. Have a look at it, and how you think you will think that it is how relevant it is today, and how we can make it more relevant. So please, I would like you to really look at it and do let us know. And secondly, we also wanting to start honors program in biotechnology as well as in microbiology, where healthcare and diagnostics should take a front stage. So we have made something, we have made some effort, rather Dr. Rathod has been working on it with Rajita and Amina and other entire team. So I would request Dr. Rathod to kindly forward that also to Nuruddin. And you have such fantastic alumni. We all have such fantastic alumni. We never thought of taking their help, only except few. So I think all the more after today's session, we will fix your date with students of KC. So, yeah, from there you have to date KC students from there and date with with the knowledge. So I think that we are going to look at, and we'll fix every month 1 or 2 sessions. Now we are becoming more greedy after listening to most of you, and especially your talk. So may I request you that you make it a practice that this is a particular day in the month I need to visit KC College from there.
Yeah, I'll try my best.
So you need to freeze that date in your diary, and I look forward to having you, Ms. Sas, again. Imagine I was just glued to my chair. I'm still there in the office, and it is 7:15. Yeah, 7:40. So it's because of you. Thank you so much, and I look forward to hear from you again.
Thank you so much.
Take care and stay safe. And when you're going visiting your lab, take care. You're very, very important to all of us, your parents and all the family members, and even to KC family too.
Thank you so much. Yeah, and I just last thing I'll say is I'm still blown away with the questions. I was just amazed at how much people were thinking deeply about this. I just wasn't mentally prepared for that. So I'm pleasantly surprised. So
all the best, Nuruddin. Take care. I'm heading on to another meeting, so I will exit. I know I should not go before Nuruddin, but I'll have to. All the best. Take care.
Yeah. Thanks again.
Thank you, ma'am. Thank you, ma'am.
Yeah, bye-bye.
Neha?
Yes,
over to Neha for the formal vote of thanks.
Thank you, ma'am. On the behalf of KC College and the Departments of Microbiology and Biotechnology, it's my honor to express our gratitude to the speaker of the day, who is also one of our prestigious alumni, Dr. Nuruddin Unchwaniwala. Thank you, sir, for taking us through your wonderful works, imparting such a vast knowledge about the viruses. Thank you for explaining many of the tedious concepts in a very simple manner. Your enthusiasm while explaining every bit of the topic greatly reflected the love for your studies in this field. I'm sure many of our participants must have been inspired by you this evening. Thank you once again, sir, for such a wonderful session. We wish you all the very best for all your future works and may you achieve great success ahead. I would also like to express our profound gratitude to our principal, Dr. Hemlata Bagla ma'am, our star status coordinator, Dr. Sagrika Damle ma'am, for their constant support and encouragement. Also, thank you, Monish, for all the technical support. Finally, a big thank you to all the wonderful participants who have turned up in such great numbers. Thank you for being such a great audience and for all your cooperation. Once again, thank you all.
Yes, so that's how we come to an end of this session. I request Prabha ma'am to please interact. And Israni ma'am. Is Israni ma'am there? Prabha ma'am? Israni ma'am has
There
joined by
she
Mita.
is.